Thanks for coming again. And I think I walked past some of you last night or the night before, and I didn't say hi. Was that you guys? Yeah, okay. Sorry I didn't say hi. I didn't recognize you at first. So we'll just quickly go through amino acid breakdown. We won't spend too much time on that. And then we'll go through oxidative phosphorylation. So amino acids can also be broken down for energy. So what that means is that if you eat or consume excess protein, or if you're starving, then your body will start to break down your muscles. So if you've ever seen someone that's starved for a long time, they'll have very little muscle, and that's because their body is breaking down the muscles so the muscles are made of proteins, and the proteins get broken down into amino acids, and then the amino acids get turned into carbon skeletons, which can then be used for energy, right? And going the other way, what that also means is that if you eat too much protein, proteins and amino acids can also be turned into fat as well. So if you think that if you just eat a lot of protein, you won't get fat, that's not true. It's harder, but if you eat enough proteins, your body can still turn it into fat as well. So in order to do that, amino acids need to have their amino group removed. So they need to be removed in order to be used for energy. And this amino group can be removed and released into solution as NH3, which is otherwise known as ammonia or ammonium. So if you have let's just say, here's your R group, and then here's your so here's your amino group. That can just come off, and your amino group comes off, and if it's floating on its own, it's ammonia, but usually if you're at a pH, at physiological pH, so that's about a pH of 7.4, most of your ammonia will get protonated and turned into ammonium. So NH4 plus, and this is ammonium. Okay, so like in your exam, it doesn't really matter too much whether you say ammonia or ammonium, but it will mostly be in that ammonium form when it's in the blood. So they can be removed and released into solution as ammonia or ammonium, and that's called deamination. So you're just taking the amino group off, or you can transfer it onto another molecule, and that's called transamination. So you're transferring it to another molecule instead of just putting it in, removing it. So ammonium in the blood, if a lot of this in the blood is toxic, so ammonia, if it gets into the brain, it'll cause, it can make you go psychotic, and that'll cause problems. So we want to minimize the amount of ammonium in the blood, okay? And so transamination is when the amino group is transferred onto an alpha keto acid, okay? So keto acid is simply a molecule so if you remember your chemistry, this is your carboxyl group, this is and the first carbon after the carboxyl group is your alpha carbon. So your alpha carbon has a double bonded oxygen, which is a ketone, so that makes it a ketone. So that's why it's called an alpha keto acid. And this requires an aminotransferase enzyme, or you can say transaminase enzyme. And the enzyme the transaminase enzyme requires pyridoxal phosphate. So I'll write that down. So pyridoxal phosphate as a coenzyme, and that's made from pyridoxine. And which B vitamin is that? 6. Yep, that's right. So that's vitamin B6. So pyridoxine. And so pyridoxal phosphate acts as a coenzyme, so it carries the amino group temporarily and then transfers it onto the alpha keto acid. And the most common transaminase reaction is transfer of the amino group onto pyruvate to create alanine. So if you think all the way back to the start of semester 2, you guys learned about amino acids. So alanine is one of your, is actually like one of the most basic amino acids, which can travel in the blood as a carrier of the amino group and then drop it off in the liver, where it can be converted into urea. Okay, so the liver is able to convert these ammonium into non-toxic, something that's non-toxic, and that's urea. Okay, and you'll just pee urea out in your urine. And that's why, so if you've ever got a liver blood test, so a blood test looking at your liver function, I'm not sure if you guys have ever looked at your results, but if you did, what they're looking for in the blood is these enzymes, so your ALT levels and your AST levels. So ALT is alanine transaminase, and so these enzymes are only supposed to be in your liver cells. So normally, if your liver is healthy, there won't be any of this enzyme in your blood, but if your liver cells are dying and damaged, then they'll release all these enzymes into your blood. And so if you take a blood test and you see lots of liver enzymes in your blood, that shows you that tells you that your liver is not healthy right now. Okay, so next time you get a blood test and see your liver results and you see ALT and AST, you know what they are. Okay, so deamination involves the amino acid being oxidized and losing its amino group. This usually only happens in the liver cells, which are the only cells capable of converting ammonium into urea. So we'll move on to the next page. So once the amino group has been removed, the carbon skeleton can be used to make acetyl-CoA, or used as substrates in the citric acid cycle. So you can see in that chart below how all of your amino acids, your 20 amino acids, enter into the citric acid cycle as those carbon skeletons, right? So, and that all gets broken down into acetyl-CoA. So I've got a couple of questions here from past exams. The role of pyridoxal phosphate in aminotransferase reactions is to A, catalyze the reaction. So is that true or false? So is pyridoxal phosphate an enzyme? No, it's a coenzyme, so it's just a carrier. So it's not going to catalyze the reaction. That's the job of the enzyme, which is aminotransferase. So B, so that should say accept an amino group from an amino acid and transfer it to a keto acid. Is that true or false? Yep, sounds right. So it's true. So that's its job, to transfer. It's going to carry that amino group. C is false. It doesn't provide oxidizing power. There's no oxidation or reduction happening in this reaction. And D is obviously false. So the next question, which one of the following statements about the human metabolism of amino acids as a fuel molecule is incorrect? So A, carbon skeletons from certain amino acids can enter into the citric acid cycle. Is that true or false? Yep, true. We just said that. B, the alpha amino group that is removed from amino acids is oxidized to produce ATP. True or false? So what happens to that amino group in the end? Yep, so it goes to the liver, gets converted into urea, and it gets peed out. So B is not true, so B is incorrect, and that's your answer. C, transamination reactions involve the transfer of an amino group from an amino acid to a keto acid. We know that's true. And D, transamination reactions are involved in the safe transfer of excess nitrogen to the liver, where it becomes part of urea. So that's true. So we'll move on. So now we're going to go through oxidative phosphorylation. Oxidative phosphorylation involves the electron transport chain, or the ETC, and ATP synthase. And the two are coupled by a proton gradient. This means that the energy required for the production of ATP comes from oxidative phosphorylation, which is indirect as opposed to substrate level phosphorylation. And I apologize because last week I didn't mention substrate level phosphorylation at all, but it is important to understand the difference between substrate level phosphorylation and oxidative phosphorylation. So I've just drawn out what I feel is the easiest way to think of it. So in substrate level phosphorylation, the substrate has high energy, so things like 1,3 BPG. So remember that was the molecule, the 3-carbon molecule, 1,3 BPG. And it had a negatively charged phosphate on the first and the third carbon. So it's high in energy because these two negative charges are going to repel. And so we use the energy in this substrate to convert ADP into ATP. So the energy comes from the substrate, and this is the substrate. Whereas in oxidative phosphorylation, the oxidation of NADH and FADH2, our electron carriers, that produces energy via the electron transport chain, and that energy is converted into the proton motive force, okay? And then the ATP synthase uses that energy of the proton motive force to phosphorylate ADP into ATP, okay? So that's the difference, and it's important. And so that means that the two processes of oxidation and phosphorylation are coupled, okay, where the energy generated by oxidation is utilized by phosphorylation. And that's an important concept to remember as well, the coupling, because when they say uncoupling, so if you have a molecule that can uncouple those two processes, that's what they're referring to. You have oxidation, but that energy isn't being converted into phosphorylation. So now the electron transport chain is located on the inner mitochondrial membrane, and is comprised of 4 protein complexes. So this one's really easy. It's called complex 1, 2, 3, and 4. And there's nothing too confusing about it, which is pretty funny considering it's like everything else seems like kind of doesn't make sense. So the electrons get passed on to carriers that have increasing reduction potential. So higher reduction potential means that they want to gain electrons more, and so they want to be reduced. And they receive electrons from NADH and FADH2. So remember that during glycolysis and the citric acid cycle, we generated NADH and FADH2. And so we extract the energy from those electrons to pump the protons across the inner mitochondrial membrane into the intermembrane space. So that's going to create a high concentration of protons in the intermembrane space. Okay, so the space between the inner mitochondrial membrane and the outer mitochondrial membrane. And the electrons are then transferred onto oxygen to form H2O. So oxygen is our terminal electron acceptor. And that's why we need oxygen. So if you don't have oxygen to take those electrons away, the whole electron transport chain would stop. And so in the diagram at the bottom of that page, I've just labeled UQ. So Coenzyme Q, so you should just call it Coenzyme Q, but it's also known as ubiquinone, and so that's why it's labeled as UQ in that diagram. But just remember that UQ is the same thing as Coenzyme Q. So NADH drops off 2 high energy electrons at complex 1, and energy is extracted from these electrons to pump 4 protons into the intermembrane space. So I like to think of these electrons as batteries. Okay, so if you were to say, compare it to a phone, the electron would be at 100% charge, right? And so we're going to take out some of that energy to pump protons against their concentration gradient, because remember that the concentration of protons is low in the mitochondrial matrix and high in the intermembrane space. So if you want to pump protons from the matrix to the intermembrane space, that's going against its concentration gradient and also against the electrical gradient as well, because protons are positively charged. So you're going to need energy to do that. So the electrons are now slightly lower in energy and are passed on to coenzyme Q, which is also called ubiquinone or UQ, and then so coenzyme Q is going to deliver the electrons to complex 3. So the reason that we need these mobile electron carriers, such as coenzyme Q, is because these complex 1, complex 2, 3, and 4 are large proteins, and they're not able to move in the membrane, as well as the smaller things like coenzyme Q. So they're able to pass the electrons on. So then complex 3 pumps 4 more protons and passes the electrons that are even lower in energy onto cytochrome C. So cytochrome C is a peripheral protein on the intermembrane side of the inner mitochondrial membrane. And then cytochrome C is going to deliver the electrons to complex 4, which pumps 2 protons. So, yeah, and then the electrons are passed on to oxygen and water is formed. So if you look at the path of the electrons for NADH, it goes from complex 1, skips complex 2, and goes straight to complex 3, and then to complex 4, and then to oxygen. And we've pumped 10 protons out for each NADH molecule. So I'll just quickly draw a diagram. So here's your complex 1. So you've got NADH, which is carrying the fully charged electrons, and it drops it off. And so NADH gets turned back into NAD+, right? And at the same, so we're going to drop these electrons off. So there's NADH is carrying 2 high energy electrons, okay? So these are high energy. And then as it goes through the complex, we use that energy to pump 4 protons into the intermembrane space. So which complex is this? Yep, complex 3. And the electrons are transferred to coenzyme Q. And these electrons are now, they've had some energy taken out of them. So now we'll just say they're medium energy. Okay. And we're going to use that energy to pump 4 more protons. So this is complex 4. And we have a protein called cytochrome C, and this is outside of the membrane. So the electrons have gotten more energy taken out of them. They get transferred to cytochrome C. Cytochrome C takes it to complex 4. And then, so we've got our electrons here now. And at this stage, so when they were here, they were at medium energy, and they've got more energy taken out of them. So now they're low energy. And then we're going to use, now it doesn't have a lot of energy left, so we can only pump 2 protons, not 4. Okay? So we're just getting the last bit of energy that we can out of those electrons. So we're going to pump 2 protons out. So now these electrons, once they get to the end of this electron transport chain, they're very low in energy, or you could say no energy, but technically that they would still have energy, but you can just think of it as having no energy or very low energy. Okay, and then you'll have you'll have an oxygen plus 2 protons. So they'll combine with the electrons to form H2O. Okay. So that's NADH. And now we'll go through FADH2. So remember, FADH2 is produced during this beta oxidation and also the citric acid cycle. So remember that succinate is oxidized to fumarate in the citric acid cycle. So if you remember last week, I said that one of the reactions didn't occur in the mitochondrial matrix, one of the citric acid cycle reactions. So this reaction actually occurs on complex 2 of the electron transport chain. Okay. And during this reaction, 2 electrons are passed onto FAD to form FADH2 and then released into the electron transport chain. So you've got this is complex 2. I'll draw it a little bit bigger. This is complex 2. You've got this is your citric acid cycle where succinate is turned into fumarate. And as part of this reaction, FAD is turned into FADH2. And then FADH2 is going to lose its electrons, and you're going to have 2 electrons come off. Now these electrons that have come from FADH2, they're not that high in energy. So if we talk about in terms of numbers, if NADH2, the electrons from NADH2, if they were 100% charged, these are only about 70% charged or 60% charged. So these only have, we'll just say, medium energy, right? So we don't have that much energy in these electrons, so we're not going to take any energy out of them. We're just going to pass them straight on to coenzyme Q. So there's no pumping protons from complex 2. And we have our complex 3. So the electrons are going to come here. And remember that we didn't take any energy out of them, so they're still going to be medium energy. Okay? And then complex 3 is going to use this energy to pump 4 protons, just like for NADH. And then the rest is all the same. So you've got cytochrome C, which acts as the carrier, 2 electrons, and by this it goes from medium energy to low energy. Okay, so we've only got a little bit of energy, so we can only pump 2 protons. And once we've extracted all that energy, they're going to be very low in energy. Okay? And then the rest is all the same. So, in terms of inhibitors of the electron transport chain, you have two compounds that you should know about. Okay? So the first one is called rotenone. So rotenone is going to inhibit coenzyme, it doesn't actually inhibit coenzyme Q. It inhibits this transfer process. So it inhibits the transfer of electrons from complex 1 and complex 2. So it's just got complex 1 here, but remember that it's only got complex 2 here, but remember that complex 1 also gives electrons to coenzyme Q. So rotenone is going to inhibit this process. And the other one that you should know about is cyanide. So cyanide is going to inhibit complex 4. So it's going to stop complex 4 from working. To be specific, it inhibits something called cytochrome A3, which is a carrier of the electrons. So that'll stop the electron transport chain. So that's all on the next page. But I don't have a diagram on the next page. So on the next page, you've got electron chain inhibitors. Cyanide binds to cytochrome A3 inside complex 4. And the inhibition of cytochrome A3 means the electrons can't move through the ETC and no proton gradient is produced. And so that means no ATP. And rotenone inhibits CoQ, or coenzyme Q, the electron carriers that delivers electrons from complex 1 and 2 to complex 3. And if the electrons can't be delivered to complex 3, then the whole electron transport chain is going to stop and no proton gradient is formed, which means no ATP. So again, I have a couple of questions. 21, which statement about the electron transport chain is incorrect? So A, the electron transport chain involves electron carriers undergoing a series of redox reactions. Is that true or false? Have a guess? Yeah, it's true. So it's just, it's electrons going from one carrier to another within these complexes. B, the electron transport chain is organized into a series of protein complexes between which mobile electron carriers transport electrons. Yep, true. So the mobile electron carriers are coenzyme Q and cytochrome C. C, the energy released during the passage of electrons in the electron transport chain is used to pump protons across the inner mitochondrial membrane. Yep, so that's true. And D, NADH is the terminal electron acceptor in the electron transport chain. Yep, false. So what is the terminal electron acceptor? Yep, that's right. Next question. Molecules such as rotenone and cyanide inhibit the electron transport chain. The consequences of such inhibition includes A, increased ATP synthesis by ATP synthase. So is that true or false? Yep, false. So remember, you're going to decrease ATP synthesis because you're not getting that oxidation, which is how you're extracting the energy from the electrons. B, an increased rate of beta oxidation. Is this true or false? Yep. So why would it be false? Yep, that's right. So I think you know why, but I'll just explain quickly to the others as well. So we have complex 1, which turns NADH into NAD+. We have complex 2, which turns FADH2 into FAD. And for beta oxidation, beta oxidation turns NAD+ into NADH, and it also turns FAD into FADH. Okay, so in order to perform beta oxidation, we need NAD+ and we need FAD. Okay, so if we have a inhibition of the electron transport chain, what we're doing is we're preventing this from occurring. So we don't have NAD+ and we don't have FAD. So that means that beta oxidation would slow down, not increase. Okay. And that's also the same for glycolysis. So if you remember, glycolysis, it turns NAD plus into NADH. Okay. And remember that sometimes if you don't have enough oxygen, that can also inhibit the electron transport chain because there's nothing to take away those electrons. And so if that's happening, so then if you don't have complex 1 turning NADH into NAD+, then you won't have any NAD+, which means glycolysis will also stop. So in anaerobic, I'm going a little bit off topic here, but it's relevant. So what the cell does, if there's no oxygen, is we need to get NAD+, right, in order to keep doing glycolysis. And this is important because otherwise we'll have no energy. So the way we regenerate NADH into NAD+, how do we do that if there's no oxygen? Yep. So we're going to transfer these transfer the electrons back onto pyruvate, and pyruvate gets turned into lactic acid or lactate. And then we have NAD+, which means we can keep performing glycolysis. Okay, so these are all these things that you're learning about are all connected. Okay, and that's what we're trying to do. We're just trying to make you understand how all of these are connected. We're learning about these one at a time, but in your exam and going forwards, you have to understand how they all fit in together and how if you inhibit one process, it's going to affect other processes. So we'll move on to the next page and we'll talk about ATP synthase. So ATP synthase is located on the inner membrane of the mitochondria, along with all your other protein complexes. So the pumping of protons into the intermembrane space creates an electrochemical gradient and a proton motive force. Okay. So those are key words that you want to remember: electrochemical gradient and proton motive force. Okay. And this means that protons want to flow back into the mitochondrial matrix where the concentration is lower. And so remember I said that the inner mitochondrial membrane is very impermeable, so things can't go through it. And so that means that the only way the only way the protons can get back into the mitochondrial matrix is through ATP synthase. And that's going to make the rotor part of ATP synthase rotate. Okay? So ATP synthase uses the flow of protons to generate ATP. So the ATP synthase can be divided into F0 or F1, where F0 is the part that's in the mitochondrial membrane. So here's your membrane. And you have this part here. So this part here is F0, and this part here is F1. Okay. And then next page. So it can also be divided into rotor or stator parts. And so the stator doesn't move, is stationary. It's called stator because it doesn't move or is stationary, while the rotor parts rotate. So there should also be, so I'll draw this in another color actually. So the parts in green is the rotor part, and the part in blue is the stator part. Okay, so the green part, the rotor, is going to rotate, and it's rotating because protons are coming through. So protons are coming through and making this green part turn, and so this is the stalk. This green part here is the stalk, and this part here. So the stalk is inside. The stalk is inside the catalytic head. So this is the catalytic head. And these here are the alpha and beta subunits. So the catalytic head is composed of alpha and beta subunits. And these alpha and beta subunits are the parts that bind ADP and inorganic phosphate and turn it into ATP. So ADP plus inorganic phosphate, they get turned into ATP in this catalytic head. Okay, so the catalytic head doesn't turn. It's part of the stator. Yeah, okay. So we'll just go through the numbers now. So 1 ATP is made for every 4 protons through ATP synthase. So if you have 4 protons go through ATP synthase, you get 1 ATP. And then if you remember that for each NADH, we pumped 10 protons. So that means that each NADH pumps enough protons for 2.5 ATP. And FADH2 pumped 6 protons. So it's going to produce 1.5 ATP. Okay, and it's important to remember those numbers. Okay, because it comes up in the final exam. So you need to be able to remember if you had a certain number of NADH and a certain number of FADH2, how many ATP you'd be generating. Okay, and you also need to remember how many NADH you get from glycolysis, from the citric acid cycle as well. Okay, so just make sure you go over that. So I'm just going to talk about dinitrophenol or DNP. So this can uncouple the electron transport chain, which is oxidation, from ATP production. So it works by picking up protons from the intermembrane space and then crossing the inner mitochondrial membrane. And so it's going to decrease the proton concentration, decreases the proton concentration in the intermembrane space, which means less protons pass through ATP synthase and less ATP is produced. But remember that we haven't inhibited the electron transport chain, right? We've just uncoupled oxidation from phosphorylation. So the electron transport chain can keep going, and in fact it's going to go faster because so normally the cell it doesn't want to keep using up NADH and FADH2 if we already have ATP. So remember the goal of the electron transport chain, glycolysis, and citric acid cycle is all to produce ATP. So if you have enough ATP, that's going to act as an allosteric inhibitor. So remember, going back to enzymes and proteins, ATP can inhibit those enzymes that are in the electron transport chain and also the citric acid cycle. So normally ATP will inhibit those and make it go slower. But if you're not producing any ATP, then there's no inhibition, so the citric acid cycle will be accelerated. And this energy is turned into heat, and overdose can result in death due to hyperthermia, which is otherwise known as overheating. Okay, so I've just got a diagram on the bottom there which shows you how DNP works, and so it's also important to remember that. It's also important to remember DNP because they like to ask that in the exam as well. It comes up pretty often. So what DNP does is it uncouples, it acts as an uncoupler. Okay. So we just finish off with a few questions. Which statement about the F0, F1 ATP synthase is correct? So A, both the rotor and the stator move as protons move down their concentration gradient. Is that true or false? Yep, that's false because the stator doesn't move. B, rotor movement causes conformational changes in the stator to catalyze ATP synthesis. Yep, so that's true. The rotor, so the stalk, the movement of it catalyzes, causes a conformational change in this catalytic head, and that produces ATP. C, the F0, F1 ATP synthase is located in the plasma membrane. Is that true or false? Well, yeah, we know it's false. So the plasma membrane means the cell membrane, but this is on the inner mitochondrial membrane. Okay. And ATP synthesis requires only the F0 part of the ATP synthase, so that's false because we need both the F0 and the F1 portion. Next question, in metabolic terms, the reaction below is an example of, so what you've got there is you've got a substrate and you've got guanosine diphosphate or GDP, and it's getting phosphorylated, right? So what do we think that is? Yep, so it's a substrate level phosphorylation because we're taking the energy from that substrate on the left and taking that energy and turning it into GTP or guanosine triphosphate. So this is the reaction in the middle of the citric acid cycle, and 1 GTP is equivalent in energy to 1 ATP. And the last question, an uncoupler of mitochondrial oxidative phosphorylation. So what's an example of an uncoupler? Yep, so DNP is an uncoupler of mitochondrial oxidative phosphorylation. So A, the inability to oxidize NADH in the electron transport chain. True or false? False, yep. So it's not inhibiting the electron transport chain, so the electron transport chain can oxidize NADH. B, it will result in an increased proton motive force. So true or false? Yep. So false. Yep, false. Because those protons are leaking out. They're leaking back into the mitochondrial matrix instead of going through, and so you'll have a lower concentration gradient. And C, increased electron transport chain activity with decreased or no ATP production. So what do you guys think? True or false? Yep, so that's true. And then D is obviously false, so a requirement for ADP. Yeah, that just doesn't make any sense. So that's it for tonight. Thank you, and I'll see you guys next week. So we'll have two more tutorials, and then that's it. All right, cool. See you guys.
Hello, Ava. How well, no one else breathes on my neck.
Yeah, A. No, remember A.
Oh.
Not A.
There's some funky bouncing shit there.